Thank you very much. So today I'll be introducing the Codetta Bio suite, which consists of the Concerto system, which has our Concerto platform, as well as our multiplex panels and reagent kits, which work together with our Prelude chip to provide the next generation of multi-omic assays in a customizable and reconfigurable platform. Access is also available through Sonata Services, our in-house custom and semi-custom offerings for labs interested in exploring the technology for their own research. There are a number of challenges in traditional PCR and immuno-PCR approaches. Single-pot multiplexing and qPCR digital PCR, where multiple detection chemistries interact, can often lead to false positives, high background from nonspecific amplification, and suppressed signals from interactions between the chemistries. In immunoassays, even good antibody pairs can suffer from crosstalk problems and onerous reoptimization whenever anything needs to change in the panel. It takes a long time to dial in the sensitivity and dynamic range that you need for each one of your assays, and so any change that's needed, if you want to add a new target or take a new target away, sometimes you're starting over from square one. It can be quite complicated. Additionally, there's problems with identification and specificity for higher plex. In probe-based paradigms, where you have a single dye molecule per target, you're often limited to the number of colors that your platform can discern in the number of channels. So that typically limits you to an upper plex of about 4 to 6 or so per assay. Sample prep can also be quite a problem. The cleanup of the target from the matrix is important to prevent inhibition of downstream amplification. But also, what can occur, even when you have a good purified, something like for a nucleic acid sample, for example, you may have a large number of non-target nucleic acids that can cause problems for you in amplification downstream. So rather than multiplexing everything in the same pot and the same tube, Codetta's approach is to use single-plex reactions that are multiplexed in space. In order to do this, we're still using traditional qPCR primers for PCR and reverse transcription PCR, as well as immuno-PCR. But it's not so much the reagents, it's how they're used. So our assays aren't sequencing. They're actually highly targeted with these primer pairs and immuno-PCR chemistries. And we use magnetic beads to do a lot of the work for us. Beads are designed for each target, and first, for each set, we encode the beads with a unique chromatic barcode. Those beads are then functionalized with an affinity reagent to capture the target. In the case of nucleic acids, we use a primer. And for proteins, we use a capture antibody. We then add a detection reagent to the beads that are specific for that analyte. For nucleic acids, it's a primer pair. And for proteins, we add a forward primer that amplifies the unique tag bound to the detector for the protein in that particular assay. The complexed beads are then added to isolated microwells where there's only a single bead per well. When the amplification occurs, the chemistry remains single-plexed. The way that we get multiplexed multi-omic assays is by doing a very large number of these in parallel, where each one is a single-plex reaction, but there are many replicates of each. Talk a little bit about the sample prep integration and the workflow. So how do you get proteins and nucleic acids from the same sample at the same time? Well, we split the sample on the bench in a parallel work stream. First, the nucleic acid sample, the aliquot for that, is digested with a proteinase and then heat denatured so that the nucleic acids can bind to the primers on the beads once they're incubated. For proteins, we dilute the sample with a protein buffer so that the beads, when we mix with them, the capture antibodies on the beads can capture the targets. After that, they're all pelleted and washed. For the nucleic acids, when you're looking for RNA targets, the reverse transcription can be performed directly on the bead. When the RNA binds to the primer, the RT can extend that primer to form the cDNA directly on the bead in a bulk reaction. For the proteins, we then label those beads with a detector that has the tags for our immuno-PCR. The samples, or the beads from each sample, can then be recombined into a single tube and then loaded on the Concerto. Up to 24 of those samples can be added per run. The Concerto prepares the chip by loading the beads in the microwells. It adds the PCR master mix that can amplify the targets from the chemistries that are already on the beads. It seals them to isolate the reactions using a sealing oil that flows through the chip. Upon thermocycling, the amplification chemistries are released. The positives are identified after the PCR thermocycling by a fluorescent dye in the solution. We then look to the color of the beads to determine the identity of each of those positives. I'd like to talk a little bit about our prelude chips, which are integral to the operation of the platform. They consist of a silicon and glass device. The silicon has etched features in it that form the microwells. There's a total of about 250,000 microwells per each sample. A glass flow cell is bonded on top of that, which separates the arrays for each of those samples into 8 lanes. Each one with 250,000 wells, or about 2 million per chip. The silicon and glass portion of the chip is encapsulated in a plastic encasement, which does a lot of the fluidic handling for us. First, there's a reagent common port where a loading buffer can be added to wet the chip. There are bead loading ports, 8 different ones that access the different flow cells. The Concerto pipettes a bead slurry into each of the ports for each of the samples. A magnet underneath the chip in the silicon pulls the beads down to the silicon surface. After that, the beads are then pulled through the chip, and they flow like iron filings across the surface of the array and fall into the wells. After that, a PCR master mix is then added to the common reagent port, followed by a sealing oil that isolates the wells. All of the waste fluid that flows from the chip goes up into a pad that's located at the back of the chip that absorbs all the waste and contains it in the solid phase. So the Codetta system combines both digital quantitation at the low end of the concentration range, as well as an analog PCR quantitation for the higher concentration targets, where there are multiple copies of target per bead. The digital signal comes from measuring all of the beads on the entire chip before PCR and after PCR, and simply counting up the positive reactions for each analyte type. For higher concentration targets where you have more than a single copy per bead, the digital signal is saturated. Because the digital signal is saturated, you can't really tell what concentration you have above a certain level. However, we're able to transition to a qPCR mode where we're monitoring the signal after every cycle of PCR across a single strip on the chip. By monitoring this, we can calculate a cycle threshold, which tells us the number of copies of target on each of the beads. Combining these signals together, we typically see about 6 to 8 logs of combined dynamic range within a panel. So without doing any sample dilutions, you can look at targets that range up to 100 million-fold in different concentrations. The Concerto system has 3 channels or colors for decoding the beads, giving a 3D cluster plot that's used to determine the bead identity. We calculate the digital PCR signal by looking at those positives, as I mentioned. We also then look at the curves for the analog qPCR trace for every sample. These are used to determine the targets per reaction. That concentration is then determined by using a reference curve, which corresponds to a signal coming from internal control beads as well. The concentrations are output as a table, which is a simple CSV file for each analyte. It reports the concentration along with a QC score, whether it's in the linear dynamic range, et cetera. So I'd like to talk a little bit about our noise canceling approach. We use primers on the beads that are specific for the detection conjugate for every protein target we look at. Because the primer is coming from the beads, they're not mixed in the PCR reactions. So if we make it the correct amino sandwich and we load it into our chip, into the isolated microwells and start our thermocycling process, the positive signal is only produced where the primer from the bead can amplify the tag. For cases of nonspecific binding, where different antibodies have bound to the beads, but they're for the wrong target and would normally cause nonspecific binding, no signal is generated because we don't have the primers that can amplify that tag. Shown on the left-hand side is an example of how the noise canceling is effective at turning off the signal. If we spike in some universal primers so you can see where all the targets are binding, you can see that there's a significant amount of off-target signal. This is still less than a single detector per bead, but it's enough in a single molecule sensitive assay to cause quite a lot of trouble. But it's not just the lower noise and better sensitivity that you get. What this also does is makes the assays modular. Shown on the left-hand side is a dose-response curve for IL-1 beta, where we've used just either the detector for just IL-1 beta We've added an additional 5 in a 6-plex or an additional 11 in a 12-plex assay. As you can see, there's really no change in the response, in the dose response. Similarly, if you look on the right-hand side, you can see that the blanks for IL-1 alpha and IL-6, as well as the other ones in the assay, they're not affected by the number of detection antibodies. And so what this means is, once you have good single-plex assays that are optimized, the detection and amplification chemistries are set. They don't interact with each other, and thus they can act as a modular unit that can be ported from one panel to another without extensive re-optimization, which is usually required by other technologies for each new panel. I'd like to show you a quick example of how this can be useful. In this case, we're looking at a multi-omic sample here. This is a human plasma sample that had some spiked protein targets, or cytokines, as well as some nucleic acid sequences for respiratory pathogens. Nucleic acid concentrations are shown across the top in copies per microliter, and the protein concentrations are shown along the bottom in picograms per milliliter. Thus, we can see the disease-causing organism as well as the host response. So we have a number of off-shelf custom multi-omic kits that are available. Love for you guys to come by our suite later on. We can talk more about those. We're also excited to talk about our new coming at the end of the second quarter of 2026, our 12-plex cytokine panel of key cytokines of interest. These can be customized by adding additional protein or nucleic acid targets, as well as work with serum, plasma, CSF, and other liquid samples. You can run 8 to 24 samples per run, or up to 72 samples per day. So please come by the Park Suite, and we'd love to talk to you about how we might be able to help you with your research.